So the topic of my talk is the pituitary tumors and succinate dehydrogenase defects, and I have nothing to declare. The sections, I have divided my talk in 3 sections. In the first section, I will briefly mention the structure and the role of succinate dehydrogenase and its association in human disease. And then I will present all the available data that support its association with the pituitary adenomas, including the new syndromic association that we termed 3PAS, that stands for pheochromocytoma, paraganglioma, and pituitary adenomas. And I will end up my talk with a few conclusions. Now, succinate dehydrogenase, or complex 2 as it is known, it is an enzyme complex bound to the inner mitochondrial membrane, and since it is in the crossroads between the Krebs cycle and the respiratory chain, it has a dual role, and it catalyzes the oxidation of succinate to fumarate and transfers electrons directly to the respiratory chain. As most of you are aware, it is composed of 4 subunits, A, B, C, and D. C and D, they are the hydrophobic components that anchor the complex to the inner mitochondrial membrane and provide binding site to the ubiquinon. The SDHA is attached to flavin adenine dinucleotide, and the binding is also the binding site for succinate, where the oxidation to fumarate takes place. And the SDHB connects the SDHA to C and D and contains 3 iron sulfur centers that mediate electron transfer from succinate to ubiquinon pool. Mutations and functional variants of SDH, I will call it from now on SDH for the sake of time, have been implicated in familial paragangliomas in Carney-Stokes syndrome, in PTEN mutation-negative Cowden syndrome and Cowden syndrome-like individuals, in renal cell cancer, and in pancreatic neuroendocrine tumor more recently. Several suggested mechanisms how the defects in succinate dehydrogenase they may lead to tumorigenesis. It has been shown that loss of function of SDH causes accumulation of succinate and reactive oxygen species. So accumulation of succinate may lead to tumorigenesis through the hypoxic response under normoxic conditions, which we call pseudohypoxia, which, as you may know, it leads to the transcription of several genes that they are related to motility, survival, growth, and angiogenesis. It also inhibits developmental apoptosis of neuronal cells. It promotes the VEGF secretion and therefore local neovascularization. And accumulation of reactive oxygen species also, it has been implicated to the tumorigenesis through hypoxic response, and may also cause oxidative damage to DNA and genomic instability. Other mechanisms, they are related to epigenetic changes. So SDH-related tumors, it has been shown that they display a hypermethylator phenotype associated with downregulation of key genes involved in neuroendocrine differentiation. It has been displayed also that SDH-deficient GISTs show a greater genomic hypermethylation compared to the KIT mutant group, and more recently, aberrant DNA hypermethylation of SDHC has been suggested as another mechanism of tumor development in carnate triad, which, as you know, the gene is yet to be identified. Another very useful tool, and particularly in the clinical settings, that can guide us to screen for the presence of succinate dehydrogenase mutation is the immunostaining for SDHB, because it has been shown that loss of SDHB immunostaining is associated with mutations in any of the SDH genes with a sensitivity of 100% and specificity of 84%. In this figure, you can see SDHB staining in SDHB, C, and D mutated tumors, whereas you can see retention of immunostaining of SDHB in VHL, RET, and NF1 mutated tumors. Also, it has been shown that immunostaining negative for SDH and SDHB indicates the presence of a germline mutation in SDHA with a sensitivity of 100% and a specificity of 97%. But, there is always but, as Papa Thomas et al. Showed, in less than 10% of pheochromocytomas and paragangliomas that are SDHB negative in immunohistochemistry, they are not associated with identifiable mutations in succinate dehydrogenase, suggesting that probably other mechanisms may cause SDH instability. Back in 2012, when I was still at Statakis group, we reported for the first time a kindred that presented with familial paragangliomas, and the index case, except from multiple paragangliomas and bilateral pheochromocytomas, presented with G8 secreting macroadenoma. This family was screened positive for an SDHD mutation, and we were able to show that there was loss of heterozygosity of the SDHD locus in the pituitary tissue, as well as decreased SDHD expression and increased HIF1 alpha expression in the lysates of this pituitary tissue. Following this report, we tried to identify other reports in the literature that they may have reported with this phenotype, the paragangliomas, pheochromocytomas, and pituitary adenomas at the same time. So we were able to identify several cases since 1952. And now the problem was that most of them were reported before 90s, where the molecular techniques became available. So no genetic screening was involved. Some of these cases, actually, when we went through in details, they most probably represented cases of multiple endocrine neoplasia. But some of them may actually represent cases of what we call the new syndromic association. And actually, it was in 2008 when Lopez Jimenez and al. They reported a case of a 60-year-old male who presented with macroprolactinoma and head neck paragangliomas, and he was found to be positive for SDHC mutations. And then, in the end of 2012, a group, the Corbonis group from Barts in London, they presented similar cases, and they showed that indeed there was an association, and then the next year, other reports in the literature followed. Prompted by all these observations and reports, we decided to study the prevalence of the SDH mutations in pituitary adenomas. So we studied 168 cases from the NIH International Registry. 146 of them were sporadic and 22 were familial. Most of them, as you can see, they were secreting adenomas. And all sporadic cases were screened for SDH mutations. However, in familial cases, we were screened for SDH when they presented with this particular phenotype. Now, here are what we found. Among the 146 sporadic cases, I have to say that patients with known mutations were excluded, of course. 3 of them, they presented with the new syndromic association. And they were screened negative, though, for any succinate dehydrogenase mutations. The rest of the cases that they did not present with this particular phenotype, we identified many reported and novel variations in SDH. Among the 22 familial cases, 4 were identified with the new syndromic association, and actually 3 of them, they harbor an SDH mutation. Within the rest of the 18 familial cases, some they found to harbor MEN1 mutations, 1 with AIP, and the rest of 10 were screen negative for all the genes. So based on these results, and when we made the analysis, we found out that SDH mutations in pituitary adenomas, it is rare. And it represented 1.8% of our studied cases. We did not identify any succinate dehydrogenase defects in the sporadic adenomas. And however, SDH mutations were identified in 75% of familial cases that presented with this phenotype, which then we decided to call 3PAS, that, as I told you, it stands from pheochromocytomas, paragangliomas, and pituitary adenomas. Now, what were the clinical characteristics of the SDH-deficient pituitary adenomas? Here you can see in this table all the 7 cases that we identified with this association. All cases, so, were within the familial setting. Most cases were GH and prolactin-secreting adenomas. They were more frequently macroadenomas. They displayed more aggressive phenotype. They were more resistant to somatostatin analogs. They more often required surgery. And the pheochromocytomas or paragangliomas, they were bilateral, multiple, with a tendency to recur. And then at the same time with our report about this new association, the Dennis et al. From the Corbonitz group, in collaboration with many centers from all over the world, they studied 39 cases, 19 sporadic and 20 familial, of pheochromocytomas, paragangliomas, and pituitary adenomas. But they moved one step further and they screened for all the genes that they are related in pheos and paragangliomas, as well as in genes that are involved in pituitary adenomas. And they also performed LOH studies and pathological studies on the available tumor samples. And they identified 19 cases with the 3PAS. 8 out of these 19 cases harbored a succinate dehydrogenase mutation. In most of them, similar to our observations, there was a family history of pituitary adenoma or pheochromocytomas, paragangliomas. They were able to show LOH in pituitary tissue of the wild-type allele in some cases. Interestingly, they identified an 18-year-old patient with an invasive mixed pituitary adenoma, and which was found to harbor a VHL mutation. And they also identified 2 patients with germline MEN1 mutations, and they showed LOH of MEN1 in the pheochromocytomas. In the rest of the 20 cases, no mutation was found, and therefore the authors suggested that probably there may be another gene involved, or maybe just a mere coincidence. Another very interesting finding in the cases that they studied was these cytoplasmic vacuoles in the SDH-deficient pituitary adenomas. Keep this in mind because I will show you later another similar finding that we saw in the SDHB heterozygote mice when we studied their pituitaries. Unfortunately, they were not able to identify the nature of these vacuoles. And then other reports from the Netherlands this time. In one of them, they reviewed all the 25 cases with succinate dehydrogenase mutations in pituitary adenomas that have been reported so far. And they found that most of these pituitary adenomas, again, they may be prolactin GH secreting or non-functioning pituitary adenomas. The age of diagnosis varies, as you can see, from 15 years to 84 years. And 15 out of these 25 cases, they presented with 3PAS. But again, not in all cases LOH in the pituitary tissue was detected. Again, from the Netherlands, another group, they studied the spectrum and distribution of all the SDH mutations that have been reported so far in all tumors. I will focus here only on pituitary adenomas. As you can see, the range of the mutations in the different subunits, it's relatively equal, with the exception of SDHD, but however, because the number is so small, we cannot really make comments, except from the fact that SDH seems to be the less involved, and that most of these mutations were missense. So prompted from all these findings and in an attempt to identify the pathophysiologic mechanism of how these mutations, they may lead to pituitary tumor formation, we decided to study the SDHP mouse that was available in our lab. Now, I have to be honest and say that we were not sure what to expect. And the reason is because although this mouse was developed in order to study in more details the tumors that they are involved in SDH mutations, actually, similar to SDHD mice, they do not develop any tumors. So we were not sure what to expect. But when we studied the pituitaries, and later on a colleague of mine, Dr. Eva Zarek, the gastrointestinal tract, we saw that actually they are not so healthy. So the first observation that we made was that the pituitaries of the heterozygote mice, they appeared hyperplastic but with intact structure. And this observation was confirmed when we calculated the total number of the cells per counting field in 12 months old mice. We were able to find that there was a significant difference between the total number between in the total number between the heterozygote mice and the wild type. There was still a difference in mice over 18 months. However, there was no significant difference there. And interestingly, we observed that there was a decrease in the total number through as the age progresses. And this could be also one of the reasons why the significant difference was lost in the older mice. Another interesting observation was these blood-filled pools that we observed in the pituitary tissues of the heterozygote mice. And similar findings have been reported in the PTTG overexpressing mice. And actually, they may be an indication that the capillaries, they may be of abnormal structure, so they are more prone to rupture. When we calculated the numbers per different pituitary tumor subtype, pituitary cell subtype, significant difference was identified only in the prolactin cells, the prolactin secreting cells, in the 12 months old mice. There was a higher number in the heterozygote mice for G8 secretin, but it didn't reach statistical significance, and no difference was found for the rest of the subtypes. Now, one thing also that we observed were these intranuclear inclusions. And we also found that there was a significant difference. Actually, they were more prevalent in the SDHB heterozygote mice. We tried to identify the nature of these inclusions, which when we studied them with the electron microscopy, they didn't really look like they were into the nucleus, but they were paranuclear. And there is a question. They looked like, as you can see, empty space. So we were not really able to determine their true nature. But one of the questions that was raised, could they represent degenerative mitochondria? And the reason why this question was raised was because when we studied the morphology of the mitochondria, we saw that in heterozygote mice, they were abnormal. So they looked either elongated or swollen with destructive crista. And similar abnormal findings were reported by my colleague, Eva Zarek, when they studied the morphology of the mitochondria of human SDH deficient paragangliomas and GIST in which you can see some of them, they presented enlarged, some of them they presented swollen, and most of them they presented like cysts. Another finding also was that the chromatin in the heterozygote pituitary cells was presented diffused as a ring shaped in the periphery of the nucleus rather than a condensed centrally located foci. And similar observation has been reported in the PTTG overexpressing pituitary cells by Donagello and al. And finally, when we looked for the HIF1 alpha expression in the pituitary cells of the heterozygote mice, as you can see compared to the wild type, it was found to be increased, and actually in some cases it was nuclear located, an indication that probably hypoxia pathway is activated. However, the important thing always is to never stop questioning. So, there is always an association between the pituitary adenomas and pheochromocytomas paragangliomas. In a very nice review paper that was published last year on the 15 years of the anniversary of the identification of the succinate dehydrogenase mutations, it was shown that in 29% of the 72 reported cases with this phenotype, they had a genetic mutation in a recognized pituitary adenoma or pheo or paraganglioma predisposing gene or variant, as you can see here, and it doesn't include only the SDH components, but also MEN1, some cases of RET and VHL. Now, in 32% of these cases, no known mutation was identified, although there was a history suggestive of the presence in the family of the presence of a pituitary adenoma or pheochromocytoma. And the rest of the 39% of the cases, no known mutations were identified, but no history was present. Another question that it has been raised is the absence of LOH in the pituitary tissue, a lack of association with the succinate dehydrogenase mutations. Indeed, it may. But haploinsufficiency for the SDH genes have been suggested in bilateral adrenal medullary hyperplasia associated with a germline SDHB mutation, which showed retention of heterozygosity. Cases of pheochromocytomas due to SDHD mutations presented without loss of the wild type allele, and also cases of paradoxical loss of the mutated allele with negative, though, immunohistochemistry for SDHA and SDHB have also been reported. So the question that we raise is, are there any additional epigenetic events? So to conclude, SDH mutations in pituitary adenomas in unselected cases are rare, and based on the reported cases, they range from 0.3 to 1.8%. 3PAS describes the association of adenomas, of pituitary adenomas and pheos and paragangliomas. It is possible that some cases that present with 3PAS negative for succinate dehydrogenase mutations may represent another gene that may be responsible, epigenetic changes, mutations in two different genes, pituitary hyperplasia due to ectopic hypothalamic hormone secretion, this is something that we should always keep in mind, and a mere coincidence. 3PAS was identified in 40 to 75% of patients with a family history. Pituitary adenomas in this association can be of multiple phenotypes, mainly macroadenomas. They are more aggressive, more resistant to somatostatin analogs, and often require surgery. Pituitaries in the heterozygote mice were hyperplastic, an indication that probably hyperplasia may be the first abnormality in tumorigenesis process as initial response to pseudohypoxia. I have to remind you here that in P19, P27, PTTG overexpressing mice, and I think in beta-catenin, the hyperplasia has also been shown. Indication that the underlying mechanism of SDH-deficient pituitary tumors may be related to pseudohypoxia are the abnormal mitochondria, the diffused chromatin, and the increased expression of HIF-1 alpha. However, more studies are required. It's difficult to make genotype-phenotype correlations due to the limited number of cases. So given that in sporadic pituitary adenoma, succinate dehydrogenase mutations are rare, genetic screening is not recommended unless they coexist with pheochromocytomas or paragangliomas, or there is a family history of pheochromocytomas, paragangliomas, in which case pituitary surveillance should be considered. And at this point, I would like, first of all, to thank the Hellenic Endocrine Society, because due to the stipend that I got from them, I went to Dr. Stratakis' lab at the NIH, where all this work that I presented took place. And this led me to my current position at King's College Hospital with my new colleagues and with Dr. Adoniadou's lab. We are trying to work on new pathways, and hopefully we will have something to present next year. Thank you for your attention.